Welcome to the deep dive. Today we are taking a microscopic plunge into one of the most critical yet often underestimated structures in all of biology, the plasma membrane.
Yeah,
You might think of it as simply the skin or, the wrapper of the cell, but that view is, well, it's just far too simple.
it really is.
This thin boundary is actually the cell's ultimate gatekeeper, its structural framework, and its highly sophisticated communication hub. It defines everything the cell is and everything it does.
Exactly. I mean, if you want to understand how a cell survives, how it responds to the world, and how it maintains its distinct chemical identity, you have to look past that simple barrier function.
So that's the mission for today's deep dive.
It is. We're focusing on two vital overarching roles that the structure of this membrane is just perfectly suited to execute. And these roles, they really set the stage for all cellular activity.
Okay, let's unpack this and get right to the foundational principles. What are those two major missions that rely entirely on the membrane?
First up, there's traffic regulation. The membrane has to precisely control every single ion and molecule moving into and out of the cell.
And why is that so critical?
It's absolutely essential because it allows the cytoplasm, the inside environment, to maintain a chemical composition that is radically different from the external environment. It essentially lets the cell create its own little world.
Its own private universe, chemically speaking. And the second mission.
Second, we have signal transduction. The membrane is studded with this highly specific machinery designed to recognize exterior substances.
Like hormones or neurotransmitters.
Exactly, or other molecular messengers. And once it recognizes them, the membrane kicks off an appropriate and specific cellular response. It's basically acting as the cell's antenna to the outside world.
So it's not just a fence, it's a border crossing, a power generator, and a communication antenna all rolled into one incredibly complex, dynamic machine.
That's a perfect way to put it.
All right, so we are going to break down this essential chapter step by step, starting with the simplest, most fundamental movements across this boundary, the spontaneous actions driven by pure chemistry, hashtag tag one, diffusion and basic transport mechanisms. Let's begin at the beginning then with simple diffusion.
The most basic movement there is.
Right. The spontaneous movement of a chemical species from high concentration to low concentration. At its core, this is just a thermodynamic drive for the universe to, randomize the distribution of that substance.
And that randomization drive is something we can actually measure. When we talk about movement across a membrane, we can quantify that tendency using the change in net free energy, or P.
So you can calculate exactly how much energy is available or required just by knowing the concentration difference.
Precisamente, just the concentration inside versus the concentration outside.
And that's the key prediction, right? Whether that movement is going to happen on its own or if it needs a push.
Precisely. If that thermodynamic value, Ag, is negative, the movement is spontaneous. It happens naturally, downhill, moving down the concentration gradient. The cell doesn't have to pay for it.
But if it's positive...
Well, if that value is positive, then energy input is required to make that move happen.
Yeah.
You're trying to push the substance into a region where it's already highly concentrated.
Pushing it uphill.
Exactly.
Yeah.
And that positive energy cost tells us we're moving into the realm of active transport.
Okay, so simple diffusion is driven by that negative free energy change. Now let's talk about a very specific and crucial type of diffusion that affects the bulk of the cell. Osmosis, the movement of water.
Osmosis is really just the diffusion of water across a semi-permeable membrane. But here's the trick. We don't usually measure water concentration directly.
Right, instead we measure what's in the water, the total solute concentration.
That's it. Water naturally moves from a region of low solute concentration, which means high water concentration, to a region of high solute concentration, where there's less water.
And this movement generates a real measurable force. We call it osmotic pressure.
We do. And what's so interesting is the sheer universality of this principle. It doesn't matter what the stuff dissolved in the water actually is.
Its chemical structure is irrelevant.
Completely. It's crucial to note that for osmotic pressure, the chemical nature of the solute just doesn't matter.
Yeah.
All that matters is the total quantity of osmotically active particles on both sides of the membrane.
Can you give us an example of that?
Sure. A red blood cell sits in near-perfect osmotic equilibrium with blood serum. But the chemical makeup inside the cell and the chemical soup of the serum outside are entirely different. But the total particle count is balanced.
And this is where that structure-function relationship becomes incredibly visible and often really clinically important. we can look at how osmotic imbalances affect cells, and red blood cells are the classic model.
They really are. So if the blood serum osmotic pressure gets too high, we call that a hypertonic solution, what do you think happens?
Water's going to rush out of the red blood cell to try and balance things.
Exactly. Water leaves the red cell and the cell shrinks and shrivels. This can happen if the body, maybe due to defective renal clearance, has high concentrations of solutes in the blood.
And the opposite if the serum pressure is too low.
If the external medium is hypotonic, water rushes into the cell. The cell swells, it balloons, and eventually it bursts, a process we call lysis.
Which is obviously very bad.
Very bad. And this is where you can really appreciate the red blood cell's characteristic biconcave donut shape. It's an incredibly flexible structural feature.
It gives it a bit of wiggle room.
A built-in safety margin, yeah. It allows them to shrink when they release oxygen and swell slightly when they pick it up,
Yeah.
all without being instantly destroyed.
Now, plant cells have a much different structural solution to this same problem. They've got that rigid cell wall.
They do. In a high osmotic medium, when water leaves the plant cell, the plasma membrane actually pulls away from that rigid wall structure. This process is called plasmolysis.
And that's what causes wilting.
It is. It reduces the structural strength of the plant. Think of how plants wilt if you over-fertilize them. The solute concentration in the soil, water becomes much higher than inside the root cells, and water just flows out.
But in the normal state, water is constantly flowing in, keeping the plant rigid.
That's right. The influx of water generates massive osmotic pressure, which is counteracted by the cell wall's rigidity. The cell becomes rigid, relying on what's basically a hydrostatic skeleton. We call that turgor pressure.
So the membrane is always permeable to water.
It is. And maybe the most fascinating experimental insight here is that even when the cell is in perfect osmotic equilibrium, so no net flow of water, if we introduce labeled water, specifically tritium, we can see that water molecules are constantly passing through the membrane at a high rate.
So it's not a static balance, it's a dynamic one.
Absolutely. The membrane is always permeable to water, even when the concentrations are perfectly balanced. ####C. Characteristics of non-ionic diffusion, figure 2.2.
Okay, that covers water, which is a small polar molecule. Now let's shift to uncharged non-ionic substances. Early experiments gave us two pretty reliable rules about how these things cross the lipid bilayer.
Rule number 1, which just screams lipid bilayer, is that permeability is directly proportional to a molecule's lipid solubility.
How oily it is, basically.
Exactly. We measure this as the partition coefficient. The more oil-soluble a molecule is, the easier it slides right through that non-polar core of the membrane.
Makes sense. And rule number 2.
Rule number 2 relates to size. For molecules that are similarly oil-soluble, permeability is inversely proportional to molecular size.
The bigger it is, the harder it is to get through.
Precisely. The larger the molecule, the slower it moves, or past a certain size, it just can't move at all. Large polymers like proteins and nucleic acids simply cannot pass directly through the membrane barrier.
And the state of the membrane itself matters too, right?
Oh, absolutely. We also see how the physical state of the membrane impacts transport. Anything that reduces membrane fluidity like low temperature or increased cholesterol content also reduces the diffusion rate of molecules through it. #D ion diffusion and the electrochemical gradient.
Okay, so when we shift from uncharged substances to charged particles ions, the calculation for movement gets a lot more complex.
It does, because the membrane itself carries an electrical charge.
Right, so now you have to think about two different forces at once.
You do. You have to combine two driving forces. The concentration gradient we've been talking about and the electrical potential gradient. Remember, the inside of the cell is typically negatively charged relative to the outside.
And that internal negative charge has a massive influence on charged particles.
A massive influence. It makes the movement of positive ions or cations into the cell thermodynamically more likely and favorable.
It's like a magnet for positive charges.
It is. It pulls them inside. And conversely, it repels negative ions, anions, pushing them out.
But ions are polar, so they can't just slide through the lipids.
Highly improbable. This movement has to be mediated by intrinsic proteins that form specific pores or channels.
And we can actually infer the size of those pores by seeing what size ions get stuck.
Yeah, if we test ions with different hydrated radii, we find the diffusion rates drop off a cliff for ions larger than about 0.5 nanometers in diameter.
So something like lactate at 0.52 millimeter is just on the edge.
Right, but glycerol at 0.62 millimeter really struggles. This observation leads us to estimate that the effective size of the general Non-gated pore is around zero point 0.6 nanometers.
Okay, so now we hit a really fundamental structural complexity inside the cell, the Donan equilibrium. This is all about trap charge.
This is a beautiful concept. Cells contain these huge essential macromolecules like proteins and nucleic acids, which are often highly charged, usually negative.
And they're trapped. They're too big to get out.
They're trapped. We call them trapped anions. And the Donan equilibrium explains that the presence of these large, non-diffusible anions creates a necessary imbalance of the small diffusible ions to maintain overall charge neutrality.
So if you have a massive amount of trapped negative proteins inside, you have to keep an equal number of small positive ions, like potassium, inside to balance that charge.
Exactly. And that is why we end up with so much more potassium inside the cell than outside. Furthermore, the diffusible negative ions, like chloride, are also pushed out by that internal negativity.
And this whole thing is what creates the cell's battery.
It is. This whole phenomenon, the presence of trapped, negatively charged macromolecules, is the theoretical basis for the interior net negative charge of the cell at rest, and that is the chemical battery that powers all subsequent electrical activity.
So the natural permeability of the membrane to these cations is pretty low, even with those tiny zero point 0.6 nanometer pores. But this permeability can be dramatically amplified by special molecules.
It can, which brings us to our next mechanism, ionophores.
What are they exactly?
Ionophores are naturally occurring, often they're antibiotics, or sometimes synthetic molecules that just dramatically increase membrane permeability to specific ions. They give researchers a crucial window into how membrane architecture facilitates transport.
Okay, so we can categorize these ionophores into three structural and functional classes. Let's start with the cation carriers.
Cation carriers, like linomycin, which is highly specific for potassium, or monosan for sodium, are these incredible structural marvels.
It's going to solve a huge chemical problem, right? How to deal with a polar ion on the inside and the nonpolar lipid bilayer on the outside.
That's the challenge. And their structure is the solution. They have a hydrophobic outer region that easily dissolves in the lipid core, and a hydrophilic center, a little cage of carbonyl groups, that faces inward to perfectly bind and escort the polar ion across.
And the function of these carriers is heavily dependent on the physical state of the membrane itself.
It is. They have to physically diffuse through the fluid lipids to transport their ion. So if the lipids are highly ordered, say if the temperature drops below the phase transition, the carrier's action is significantly impaired.
Because it just can't move.
It slows to a crawl.
Okay, the second class are the proton carriers.
These are typically lipid-soluble weak acids, like FCCP. Their function is highly dependent on a pre-existing pH difference across the membrane. The molecule picks up a proton on the high concentration side and releases it on the low concentration side, effectively dissipating the proton gradient.
And the third class avoids that fluidity issue entirely. The channel formers.
Right. Channel formers, like gramoside A, are composed of long chains of hydroxylic amino acids that literally span the entire lipid bilayer. They create a stationary permanent pore that's about 0.4 to 0.7 nanometers wide.
And because it's static, it doesn't care about fluidity.
Exactly. Its action is independent of membrane fluidity and continues unabated even below the phase transition temperature.
It's incredible that these natural molecules have become such essential tools for studying fundamental cell processes.
They're indispensable. Disrupting electrochemical gradients with ionophores was central to establishing the theory that these gradients drive ATP synthesis in mitochondria and chloroplasts.
And there's clinical potential, too.
Oh, absolutely. On the clinical side, studies show real potential for drug development. For example, the calcium ionophore X537A, when tested, significantly raises cardiac calcium levels.
Which would lead to a stronger heart contraction.
Greater heart muscle contractility and increased blood flow, yeah. It offers a potential new avenue for treating heart disease.
Okay, so now let's move from these natural antibiotics to the cell's own native machinery for high-speed ion movement, the gated channels. And this story is really rooted in the groundbreaking work of Hodgkin and Huxley back in the 1950s.
Their experiments on the giant axon of the squid were just foundational. They found that a nerve cell at rest was polarized negative inside.
And when it was stimulated,
When stimulated, the membrane transiently depolarized because sodium rushed in at this incredibly fast, almost explosive rate. And that was followed almost immediately by potassium, leaving to restore the original polarity.
the critical moment of realization was the speed. They couldn't explain the currents based on any known diffusion rate or a simple carrier binding.
The numbers just didn't add up. That realization led them to propose the channel hypothesis, that excitable membranes must possess these highly specialized, naturally occurring channels that act like high-speed molecular
With
tunnels,
a gate.
with a gate that voltage or chemical binding can unlock.
But how do you even study something that small and that fast? You can't just stick a big electrode into a membrane and measure the average current across millions of channels.
You need a technical breakthrough, and that was the patch clamp technique. Developed much later, this technique overcomes the limitations of those earlier experiments.
How does it work?
Scientists use a tiny glass electrode, about 1 micrometer in diameter, to isolate a very small patch of membrane with gentle suction. This isolation allows them to measure the electrical potential difference and, in favorable circumstances, examine the properties of a single channel opening and closing.
So now we can really zoom in and see the structure. These are intrinsic proteins spanning the bilayer. What determines whether the gate is open or closed?
The proteins exist in a dynamic equilibrium between two configurations, open and closed. Gating is simply the mechanism that shifts this equilibrium toward the open state.
And that shift can be triggered in a few different ways.
Right. Gating can be controlled by voltage, as in neurons, by the binding of a ligand, like a neurotransmitter, or even by physical sense stimuli.
The voltage-gated channels in the neuron axon are the perfect example of that speed and precise control.
They are exquisitely tuned. If you look at the sodium ion channel, it's believed to have two gates, an exterior gate and an interior gate.
Okay, so what's happening at rest?
At the resting potential, the outer gate is closed. When depolarization occurs, that massive voltage shift causes the outer gate to open and sodium rushes in. That's the action potential firing.
But then it has to stop just as quickly.
Crucially, yes. Almost instantly, the inner gate closes. This establishes a mandatory refractory period where the neuron cannot immediately fire again, which prevents runaway signals.
We've learned a ton about the molecular structure of that sodium channel. Where is the actual voltage sensor located?
The channel is a very large protein, structured with 4 homologous domains, each having 6 membrane-spanning regions. The key is the S4 region within each domain.
What's so special about S4?
It's structurally unique. It contains a sequence of positively charged amino acids interspersed with hydrophobic residues. This positive charge makes it highly sensitive to the electrical field across the membrane.
And researchers confirm this role using genetic alteration.
Absolutely. When they genetically altered the S4 region to replace those positive charges with neutral ones, they effectively dampened the sensor.
So it took more voltage to open the channel.
Exactly. They found that the amount of voltage needed to open the channel increased, and the magnitude of that increase was directly correlated with how much positive charge they removed. It was definitive proof that S4 is the voltage sensor.
Beyond sodium, potassium and calcium channels are also vital in the nervous system.
Oh, for sure. Potassium channels are essential modulators. They were famously isolated based on a mutation in the Shako fruit fly. The fly trembled because its potassium flow was defective.
And neurotransmitters can affect them?
Some neurotransmitters, like acetylcholine, suppress specific potassium channels. If you suppress the exit of positive ions, the inside of the cell becomes more positive, meaning the neuron is then more likely to reach the threshold and fire an action potential.
And calcium channels are the key to the final step of communication releasing the neurotransmitters.
That's their primary role at the synapse. When the electrical signal arrives, the calcium channel opens, the ion rushes in, and that influx stimulates the vesicles to fuse with the membrane and secrete their neurotransmitters.
And outside the nervous system.
Beyond the nervous system, calcium channels are also crucial for muscle contraction. They open in cardiac and smooth muscle cells, increasing intracellular calcium, which is the trigger for contraction.
And this is where we see that direct, elegant pharmacological intervention, particularly for high blood pressure.
Yes. Hypertension drugs like verapamil and nifedipine are classic examples of channel blockers. They work by blocking these calcium channels from opening.
So you get less calcium influx.
You get less calcium influx into the heart muscle and the smooth muscle of the blood vessels, which leads to relaxation, reduced force of contraction, and a subsequent lowering of blood pressure.
Okay, moving to the second major type of gating, ligand-gated channels. The nicotinic acetylcholine receptor, found at the neuromuscular junction, is the blueprint for this action.
It is. Acetylcholine binds to the receptor, acting as the ligand, and this binding immediately opens the channel, allowing cations like sodium to flow in and causing immediate depolarization of the muscle cell.
And the receptor itself is a pretty complex structure.
It's a marvel, composed of 5 polypeptide subunits that form a central charged pore.
How does that pore maintain selectivity for positive ions?
Structurally, it's lined with negatively charged amino acids. These negative charges confer selectivity for positive ions, allowing castications to pass while excluding anions.
But the consequences of a defect in a channel like this can be devastating, which brings us to cystic fibrosis.
The CFTR channel failure is one of the most poignant examples of a single molecular mistake causing systemic illness.
It really is.
In the epithelial cells lining the respiratory tract, there's a ligand-gated chloride channel called CFTR that normally opens in response to cyclic AMP.
And when it opens,
When it opens, chloride leaves the cell, and critically, water follows. This ensures the airway is lined with moist, thin mucus that can be cleared. In CF patients, a single amino acid is missing in the channel protein,
one single amino acid.
just one. And this single deletion renders the channel insensitive to CAMP. It just doesn't open properly.
The result is immediate and catastrophic for the lungs.
The lack of chloride movement means water doesn't follow, leading to very thick, dry mucus. This creates an ideal environment for pathogenic bacteria, resulting in repeated severe respiratory infections and a significantly reduced lifespan. It affects about 1 in 2,000 Caucasians, all traceable back to a defect in this one gate.
Finally, let's just touch on sense-stimulus-gated channels, the transducers for our environment.
These are essential for our sensory inputs. For example, sound waves displace the cilia in auditory hair cells, and that physical displacement literally pulls open potassium channels, leading to depolarization and signal transmission.
And for smell.
Similarly, in the olfactory epithelium, odorant molecules bind to receptors, opening ion channels and triggering the sense of smell. These mechanosensitive channels are present in many cell types, simply responding to physical pressure or stretch.
We've established the rapid, high-traffic gates, the channels. Now we shift our focus to more selective, lower-speed transport mechanisms, the carriers. Carrier-mediated facilitated transport is crucial for getting large, essential nutrient molecules like sugars and amino acids into the cell, always moving down the concentration gradient. Hashtag A. Carrier-mediated facilitated transport, figure 2 to 8.
Think of carriers not as open turnstiles, but as highly specialized, selective customs desks.
That's a good analogy.
These are specific proteins that bind the molecule, they undergo a conformational change to shuttle it across, and then they release it on the other side. And they share some really striking behavioral similarities with enzymes.
Okay, what are those key enzyme-like characteristics?
Well, they exhibit absolute substrate specificity. They only bind one type of molecule. They show saturable kinetics, meaning if you have a huge amount of substrate, the rate plateaus because all the carriers are busy.
They're all occupied.
Right. And they're also subject to competitive inhibition by molecules that structurally resemble the substrate. This saturable kinetic behavior is what fundamentally separates carrier transport from simple diffusion, which just increases linearly forever with concentration.
And the glucose transporters are a prime example of this carrier specificity and necessity.
They're highly tissue-specific. The human erythrocyte glucose transporter, for example, is related to the one in the liver, and both play a major role in regulating blood glucose. But then you have the crucial insulin-responsive glucose transporters found in skeletal muscle and fat cells.
And in diabetes type 2, the system is compromised in a very structural way.
In insulin-resistant cells, we see a massive reduction, at least 50%, in the number of active glucose transporters present at the plasma membrane.
And that's because the cell just isn't making as many?
The reduction is linked to a corresponding drop in the messenger RNA that codes for the transporter. The cells simply cannot clear the glucose from the bloodstream efficiently, which causes dangerously high blood glucose levels.
The carrier system isn't just compromised by disease, it can be hijacked too.
The malaria parasite, Plasmodium falciparum, provides a perfect example of evolutionary theft. About 6 hours after it invades a red blood cell, the parasite synthesizes and inserts new, highly efficient carriers for carbohydrates and ions into the erythrocyte membrane.
So it can steal nutrients from the host.
To steal essential substrates from the host's bloodstream to fuel its intense replication cycle. Interestingly, the antimalarial drug fluorizin appears to exploit these new parasite-induced pathways, entering the cell and inhibiting multiplication.
Okay, so we've covered downhill transport diffusion and facilitated transport, both spontaneous. Now we face the true thermodynamic challenge, active transport, where substances move against their concentration gradient.
This is where the cell has to pay for the movement.
And the payment can be massive.
It can. Consider the parietal cells in your stomach that secrete hydrochloric acid. Inside the cell, the pH is about 7, but the gastric juice outside is pH 1.
That's a huge difference, a million-fold concentration difference for hydrogen ions.
We are looking at a hydrogen ion concentration ratio of 10 to the 6 to 1.
That's fighting a headwind of incredible force.
It requires staggering energy input. To maintain a gradient of that magnitude, the cell needs to hydrolyze the energy equivalent of many ATP molecules just to pump out one single hydrogen ion. It's why active transport is the single most energetically demanding activity for a cell.
How much energy does it consume?
Typically 30 to 60% of a typical cell's total energy budget. #C. Co-transport. Secondary active transport, figure 2 to 9.
Since pushing molecules uphill costs so much energy, the cell often tries to be clever and couple two movements together. This is co-transport or secondary active transport.
This is the cell harnessing a downhill ride to power the uphill struggle. The energy released from the favorable diffusion of one substance, the driver, which is typically sodium, is used to power the movement of a second substance, the driven, like glucose against its gradient.
And it's all managed by a single protein.
Critically, yes, the entire process is managed by a single protein carrier.
The sodium-glucose symport in intestinal epithelial cells is the classic demonstration of this coupling.
It is. The intestinal lumen has a high sodium concentration, so sodium flows readily into the cell where its concentration is low. This massive downhill sodium movement provides the precise energy required to drive glucose uptake into the cell, even when the cell already contains a high glucose concentration.
And that carrier protein is a big one.
It's an incredibly structured protein, spanning the lipid bilayer 11 times.
Co-transport is also vital for volume regulation, often seen in the sodium-potassium chloride co-transporter.
This system is critical for maintaining cell volume integrity. If cells start to shrink due to high external osmotic pressure, this transporter is immediately induced to bring in the 3 ions, and water follows, restoring cell volume.
And this system is heavily used in the kidney.
Very heavily, to reabsorb ions back into the bloodstream.
Which means we can interrupt this process pharmacologically to treat disease.
That's the mechanism of major diuretics like furosemide. To treat conditions like congestive heart failure, which involve excess fluid volume, furosemide inhibits this kidney co-transporter.
So less water is reabsorbed.
By decreasing the reabsorption of ions in water, it ensures more water is excreted in the urine, effectively reducing blood volume and pressure. #D, ATP-driven pumps, primary active transport.
When the energy source is the direct hydrolysis of ATP, we call it primary active transport. And one of the most urgent and clinically relevant examples is the multidrug transporter, or P-glycoprotein, PGP.
The PGP story is fascinating and incredibly frustrating for cancer clinicians. They observe that tumors resistant to one cytotoxic drug were often resistant to many other chemically different yet hydrophobic drugs.
And these resistant cells had huge amounts of this one protein.
Massively increased amounts of this 170 kilowatt membrane protein, yeah.
So this protein acts like a perpetual ejector seat for therapeutic drugs.
That's a perfect analogy. PGP is a membrane machine, composed of 2 copies, each with 6 membrane-spanning domains. Hydrophobic drugs diffuse into the cell, bind to PGP, and this binding immediately triggers ATP hydrolysis.
And that energy pumps the drug out.
The energy released drives a massive conformational change that physically pumps the drugs right back out. It's a relentless efflux mechanism that makes chemotherapy ineffective.
And we still don't know how it recognizes so many different drugs.
How a single glycoprotein recognizes such a structurally diverse group of hydrophobic drugs remains a mystery of its structure. It's one of the great open questions in cell biology and oncology.
All right, now let's talk about a pump that is absolutely essential for life. The very foundation of the cell's electrical potential. The sodium pump, or sodium-potassium ATPase.
This pump maintains the chemical battery that powers everything else, keeping sodium high outside and potassium high inside.
It's absolutely crucial. The concentration gradients it maintains are huge.
They are. Sodium is 15 times higher outside and potassium is 28 times higher inside. The pump uses the hydrolysis of one ATP molecule to move 3 sodium ions out and 2 potassium ions in.
And since it's moving 3 positive charges out for every 2 it brings in, it's electrogenic. It creates a net movement of positive charge out of the cell, which contributes directly to the cell's resting membrane potential.
And it's a structurally demanding protein, requiring a specific environment to function.
Yes, it needs to be integrated into a fluid membrane. And surprisingly, specific boundary lipids, especially cholesterol, are essential for its function.
It works through a precise, coordinated four-step mechanism involving phosphorylation and conformational shifts.
Right, between two key states, E1 and E2.
Okay, let's trace those conformational steps that define the cycle.
Step 1. In the initial E1 conformation, facing the inner surface, the enzyme binds sodium and a phosphate group from ATP.
Then what?
Step 2. This complex releases that sodium to the outside as it changes conformation to the phosphorylated ETP state.
Then it picks up potassium.
Step 3. E2P then picks up potassium from the outside and releases the phosphate group, becoming E2.
And the final step gets it back to the beginning. Step 4. The potassium-bound complex releases the potassium to the inside, finally returning the enzyme to the initial E1 conformation, ready for the next cycle.
Considering its critical role, it's no surprise that this pump is responsible for nearly a third of the ATP consumed by a typical mammalian cell.
It's a huge energy hog.
But its inhibition can be a powerful therapeutic tool, which brings us to cardiac glycosides.
These are plant steroids like ouabain and digoxin, isolated from the foxglove plant. For centuries, extracts were used to treat heart failure.
And they work by inhibiting the pump.
They inhibit the pump by binding to the outer surface and preventing potassium pickup. They basically jam the step 3 mechanism.
So if the pump is jammed, sodium builds up inside the cell. How does that translate into a stronger heart contraction?
This is a beautiful secondary effect. The increased intracellular sodium can't be cleared as efficiently, so it leads to a secondary increase in intracellular calcium via a separate sodium-calcium carrier system.
And calcium activates contraction.
Since calcium is a potent activator of contractile proteins, this increase results in significantly increased myocardial contractile force, helping to treat congestive heart failure. #D calcium and proton pumps.
Let's discuss two other critical ATP-driven pumps, starting with the calcium pump, which is essential for muscle relaxation.
The calcium pump is found heavily in the sarcoplasmic reticulum, and its job is extreme concentration reduction.
How extreme?
During muscle contraction, calcium concentration is about 10 micromolar. To achieve relaxation, this concentration has to drop 100-fold to 0.1 micromolar, and it has to do it in milliseconds.
That's a huge gradient to work against.
This massive calcium sequestration is achieved by the pump pushing 2 calcium ions into the reticulum per ATP hydrolyzed, working strongly against a concentration gradient.
And this pump is structurally overwhelming in the reticulum membrane.
It accounts for about 60% of the total membrane protein in the sarcoplasmic reticulum. And functionally and structurally, it shares many fundamental properties, specificity, subunits, mechanism with the sodium pump, suggesting a deep evolutionary link.
The final major pump is the H+ ATPase, or proton pump, which has diverse roles across the cell.
The proton pump is a real workhorse with 3 key jobs. First, it's critical for chemiosmotic ATP synthesis in mitochondria and chloroplasts. Second, a different type maintains cytoplasmic pH at the plasma membrane and is involved in cell wall elongation in plants. And third, and critically for endocytosis, it acidifies the interior of various cellular organelles like lysosomes, vacuoles, and coated vesicles.
What's truly fascinating is that all these diverse tissue pumps, sodium, potassium, calcium, and proton, despite their different functions, seem to belong to the same deep family.
The evolutionary homology is remarkable. They're all similarly sized, about 1,000 residues, share roughly 20% sequence identity, and have a similar architecture for their membrane-spanning regions and ATP binding site.
So they likely came from a common ancestor.
Scientists believe they descend from a common ancestor, perhaps the ancient potassium ATPase found in primitive bacteria like Streptococcus faecalis. #tags genetics of transport, clinical focus.
Since all these carriers and pumps are complex proteins coded by genes, defects in these genes naturally lead to inherited diseases. A key area where these defects manifest is in the kidney's reclamation system.
When small molecules are filtered out of the blood by the kidney, they have to be reclaimed by facilitated transport carriers before they're excreted. If these carriers fail, the essential materials are lost.
Like in cystinuria.
Take cystinuria, a recessive disease affecting 1 in 14,000 live births.
What's the functional defect there?
It's the failure of a specific kidney-facilitated transport protein designed to reabsorb 3 crucial amino acids: cysteine, lysine, and arginine.
And cysteine is the problem.
Cysteine is the least soluble of the common amino acids. When excessive amounts remain in the renal tubule because the carrier failed, they precipitate and form dangerous insoluble crystals.
Leading to kidney stones.
Painful kidney stones that can block the urinary tract, yeah.
So the treatment focuses on mitigating the consequence rather than fixing the pump itself.
Exactly. The treatment involves increasing the solubility of the cysteine, using a low-protein diet and drugs like penicillamine, which complexes with cysteine to form a more soluble dimer. It really illustrates how a simple failure in a single transport protein can have systemic, painful consequences.
Okay, moving from individual molecules and ions to large substances, we encounter bulk transport. Here, large materials don't traverse the bilayer directly, but are carried in massive membrane-bound vesicles. We're going to focus on endocytosis, the process of bringing things into the cell. Hashtag tag B, phagocytosis, eating.
Phagocytosis involves the uptake of large particles, defined as anything greater than zero point 0.2 micrometers in diameter. The membrane extends outward, tightly wrapping around the particle.
And it's a very snug fit.
Critically, yes. This forming vesicle tends to exclude most of the surrounding extracellular fluid.
This is how single-celled organisms like amoeba ingest food, but it's a critical function in mammals for our immune system.
Oh, constantly. Mammalian macrophages perform phagocytosis constantly to clear inert particles, engulf bacteria, manage antigen-antibody complexes, and clear away dead cells.
The cleanup crew.
The cleanup crew, exactly. Macrophages lining the liver sinusoids and lung airways are highly selective cleaners, helping maintain sterility, though the exact nature of how they recognize unwanted materials is still under investigation.
Hashtag tag tag C pinocytosis, drinking.
Smaller particles and bulk extracellular fluid are taken up via panocytosis, or drinking.
Mm.
This process is less selective than phagocytosis and is seen extensively in the endothelium that lines our blood vessels.
Yeah, if you look at electron microscopy images, you'll see endothelial cells are often just filled with small, non-coated vesicles, about 70 nanometers in diameter. These small vesicles are constantly pinching off and fusing moving materials across the cell in bulk.
And there is an intriguing clinical detail associated with panocytosis in diabetic patients.
There is. Normally, essential blood proteins like serum albumin are too large or the wrong shape to be internalized efficiently by these pinocytotic vesicles. But in diabetic patients, that changes.
Yes. If albumin becomes glycosylated, that is, modified by glucose, which happens non-enzymatically in diabetic patients due to chronically high blood glucose, it is then taken up by the endothelial cells.
And this is bad for the capillaries.
This aberrant pinocytosis of glycosylated albumin is hypothesized to contribute directly to the capillary damage frequently observed in diabetic patients. Hashtag tag tag DD, receptor-mediated endocytosis, coated pits.
While general pinocytosis is a bit like bulk sampling, the cell has an incredibly specific, high-priority mechanism for large molecule uptake. Receptor-mediated endocytosis, which often involves specific membrane invaginations called coated pits.
This specific uptake was first observed in developing mosquito oocytes selectively taking up yolk protein. The pits are lined on the cytoplasmic side by a dense, fuzzy layer composed of multiple units of the protein clathrin.
Clathrin, that's what gives it the coat.
It gives the forming vesicle its characteristic geometric basket-like coat.
So how does this ensure that only specific cargo is internalized?
Specific ligands bind to their receptors, which are diffusing randomly on the cell surface. These receptor-ligand complexes then migrate laterally until they reach a coated pit.
But once they're there, they're trapped.
Once there, they become immobilized and are pulled inward as the pit pinches off to form a vesicle.
The receptor itself needs a specific internal signal, a sort of docking tag, to initiate that immobilization.
Exactly. This tag involves adapter proteins, or adaptins, on the cytoplasmic side. Adaptins interact with short 4-6 amino acid domains on the receptor's tail, domains that form a sharp structural turn. This interaction ensures that only the ligand-bound receptors are pulled into the pit and internalized.
The sheer speed of this membrane turnover is staggering.
It's phenomenal. Studies on fibroblasts show they internalize the equivalent of their entire plasma membrane every single hour.
Every hour. That's unbelievable.
And because the cell retains its size, there must be continuous rapid recycling and addition of new membrane to replace what was internalized.
And what's the immediate fate of the internalized cargo in the receptor?
Once inside, the vesicle sheds its clathrin coat, becoming an endosome. The endosome immediately acidifies its lumen using a proton ATPase. This acidic environment causes the ligand to dissociate from the receptor.
So the receptor gets reused?
The receptor is then recycled back to the plasma membrane. It's a rapid 15-minute round trip, while the ligand is transported to the lysosome for degradation. This sophisticated shuttling ensures the cell reuses its expensive receptor machinery, minimizing resource waste.
One of the most important clinical examples of this receptor-mediated pathway involves low-density lipoprotein, or LDL, the primary carrier of cholesterol in the bloodstream.
LDL uptake is handled entirely by this pathway. And once the cholesterol is released in the lysosome, it performs a crucial feedback function. It inhibits the cell's own endogenous cholesterol synthesis. This maintains stable, healthy cellular cholesterol levels.
But when the LDL receptor is defective, we see the severe consequences of familial hypercholesterolemia, or FH.
FH affects about 1 in 500 people. Patients have dangerously high blood LDL levels, which leads to cholesterol deposits inside arteries, atherosclerosis, causing heart attacks tragically early in life.
And researchers identified several classes of non-functional LDL receptors based on genetic defects, showing how structure dictates disease.
Exactly. So the mutation dictates precisely where the defect manifests in the uptake process.
What are some examples?
Well, some receptors have a deletion in the extracellular domain, meaning LDL can't bind at all. Others bind LDL perfectly, but have a deletion in the bilayer spanning region, so the entire receptor complex can never be pulled inward.
And a third type.
A third type has a critical mutation in the cytoplasmic tail, the part that interacts with the adaptins, which prevents the receptor from ever entering the coated pit.
But the result is always the same.
In all three cases, the lack of internalization means blood cholesterol remains high, and the cell just continues to synthesize its own cholesterol unabated.
This internalization mechanism also applies to how the cell handles peptide hormones, like insulin, though with a key difference from LDL.
Peptide hormones are internalized only after they bind to their receptors. This is a contrast with the LDL receptor, which is constantly internalized and recycled, whether it's bound to LDL or not.
And this internalization might be important for the hormone's effects.
It may be key to the hormone's long-term effects, yeah. And defects in this process are one of the hypotheses for the decreased receptor numbers we see in insulin-resistant diabetes.
Okay, so the second major mission of the plasma membrane, beyond controlling traffic, is recognition and signaling. This governs how the cell reacts to indirect chemical messengers from the environment.
Right. Peptide hormones, like insulin, are too large and polar to cross the bilayer, so they must use specific cell surface receptors. This is the definition of signal transduction for these messengers.
Whereas steroid hormones can just slip right through.
Exactly. Hydrophobic hormones, like steroids, are nonpolar and easily slip into all cells. Receptor differentiation for them occurs intracellularly in the cytoplasm or the nucleus, #A, hormones and cell surface receptors, figure 220.
So once a peptide hormone binds, it can exert its effects directly by linking to a pump, or indirectly by triggering a whole cascade.
In a direct action, the receptor itself is or links immediately to a transporter. For example, the plant hormone auxin induces a proton ATPase pump at the plasma membrane.
And what does that do?
Pumping protons into the cell wall loosens the wall structure, which allows the cell to elongate rapidly in response to the hormone.
But the indirect scheme is far more complex and involves generating a powerful internal amplification signal, a second messenger.
That's where molecules like cyclic AMP or CAMP come in, take epinephrine, adrenaline. When it binds to its receptor on an adipose cell, it stimulates an effector protein, adenylate cyclase, located on the intermembrane surface. This enzyme then catalyzes the synthesis of CAMP.
And CAMP kicks off the response sequence, acting as a powerful amplifier.
Absolutely. The CAMP activates a protein kinase. The kinase then phosphorylates another enzyme, lipase, turning it on. And the active lipase then hydrolyzes stored lipids, releasing fatty acids for energy.
So one hormone molecule leads to many KMPs, which leads to many activated enzymes.
Right. The overall pathway shows the amplification. Hormone binds, you get CAMP production. CAMP activates kinase, kinase phosphorylates target proteins, and you get a massive physiological change. #tag B, the G protein cycle, figure 222.
This indirect signaling often relies on a complex molecular relay known as the G protein cycle. It's a core mechanism that uses three crucial proteins: the receptor, the G protein, and the effector.
The cycle's efficiency relies on the G protein's ability to switch between two states. We can trace the 5 distinct steps.
Let's walk through them. Step 1.
Step 1. Ligand binds to the receptor, causing a conformational change that allows the receptor to bind the G protein, which starts out bound to GDP.
Okay, so now the G protein is activated.
Step 2. The receptor-G protein interaction forces the G protein to eject GDP and swap it for the higher energy molecule GTP. The ligand is then released.
Step 3.
Step 3. The activated G protein, now bound to GTP, diffuses laterally through the membrane to activate the effector protein, like adenylate cyclase.
Step 4 is the action.
The effector starts its function. And then step 5, crucially, the G protein has intrinsic GTPase activity.
A self-destruct switch.
A self-turn-off switch, yeah. It catalyzes the hydrolysis of GTP back to GDP. This inactivation causes the G protein to dissociate from the effector, turning the system off and returning it to its initial state.
This system allows for immediate sensitive regulation, and adrenergic receptors are great examples of how G proteins can either stimulate or inhibit a pathway.
Yeah. Beta-adrenergic receptors, which have 7 membrane-spanning regions, use a GS or stimulatory protein to activate adenylate cyclase and increase KMP,
Yeah.
often leading to muscle contraction.
With the alpha receptors do the opposite.
Conversely, if epinephrine binds to an alpha-adrenergic receptor, a different G protein, G or inhibitory, acts to inhibit adenylate cyclase, achieving the opposite, inhibitory effect.
And nature has perfected toxins that exploit this regulatory cycle, turning the switch permanently on.
This is perhaps the most vivid structure-function failure. Cholera-toxin modifies the G's protein by adding a chemical group, poly-ADP ribose.
And what does that do?
This modification permanently prevents the G's protein from executing its intrinsic GT-PACE activity, that crucial self-turnoff switch. The G's protein becomes locked in the always active position, continuously stimulating adenylate cyclase.
And the result is a massive, uncontrollable physiological response.
Exactly. The excessive constant level of intracellular KMP causes massive water and electrolyte release in the intestine, leading to severe, life-threatening diarrhea and dehydration. Pertussis toxin acts similarly, but targets other G proteins, like G, with similar runaway consequences.
The G protein cycle isn't just for hormones, it's fundamental to our senses too, particularly vision.
It is. In the eye, when light is absorbed, the pigment rhodopsin is activated. Rhodopsin activates a specific G protein called G, or transducin.
And that activates an effector.
GD then activates the effector CGMP phosphodiesterase, which rapidly breaks down CGMP. Normally, CGMP keeps sodium channels open in the dark.
So when light hits, the channels close.
The CGMP level plummets, the channels close, the cell hyperpolarizes, and that electrical signal is sent to the optic nerve.
Beyond KMP, the GP protein cascade generates entirely different second messengers using phospholipase C.
Right. GP activates phospholipase C, which cleaves lipids in the membrane to produce 2 major second messengers simultaneously.
What are they?
The first is inositol triphosphate, or IP3, which is soluble and quickly causes calcium efflux from vesicles and the endoplasmic reticulum. This internal calcium surge stimulates critical processes like secretion and cell division.
And the second one?
The second is diacylglycerol, DAG, which remains membrane-bound and activates protein kinase C, or PKC. PKC then phosphorylates a host of target proteins, controlling processes like ion channel function and gene transcription. #TechC Rhys proteins in cancer.
Speaking of GTP activity, the Rhys proteins are a major focus in cancer research because of their similarity to the G protein system.
Rhys proteins, located on the cytoplasmic side of the plasma membrane, are structurally analogous to G proteins. They bind GTP and they possess GTPase activity.
But their link to disease is very clear.
Very clear. Their exact mechanism of action is still being determined, but we know why they're so prevalent in oncology.
Which is?
In many human cancers, including bladder cancer, endogenous Rhys genes are mutated. Like the G's protein modified by cholera toxin, this mutation often prevents the Rhys protein from hydrolyzing GTP.
So it's stuck in the on position.
It creates an always active RAS protein that continuously signals for cell growth and division, fundamentally contributing to the cancerous state. Detecting this mutation can be used to identify tumors and underscores how a failure in a single hydrolysis switch can drive proliferation. #D growth factors and tyrosine kinase receptors, figure 224.
The other major non-G protein signaling mechanism involves the tyrosine kinase receptors, or TRKs, with the insulin receptor being the classic model.
The insulin receptor is typically a complex protein with 2 parts, an extracellular subunit for hormone binding, and a transmembrane subunit that has a cytoplasmic domain with intrinsic tyrosine protein kinase activity.
So the signal isn't relayed by a separate G protein, the receptor itself is the enzyme.
Exactly. The binding of insulin immediately activates this internal kinase. That activation is essential for the immediate physiological effects, like increased glucose transport.
How does it work?
The kinase phosphorylates target proteins on their tyrosine residues, thereby altering their function. If a cell's receptors lack this intrinsic kinase activity, insulin has virtually no effect on glucose transport.
And these receptors possess a mechanism of self-regulation.
They do. Receptors often phosphorylate themselves, a process called autophosphorylation, which is believed to modulate their activity and initiate the process of downregulation. This desensitization ensures that prolonged hormone exposure doesn't lead to overstimulation.
Often by just having fewer receptors on the surface.
Right, via endocytosis.
And the link between these receptors and oncogenes is direct and profound.
It is. Growth factors stimulate cell division, and many use TRKs. Certain viral oncogenes, like VRBB, were found to be highly homologous to growth factor receptors, specifically the epidermal growth factor receptor.
But with a key piece missing.
Crucially, the viral oncogene often lacks the ligand-binding domain.
Meaning the system is always firing, regardless of whether a growth factor is present.
Precisely. Lacking the external regulatory domain means the cytoplasmic tyrosine kinase activity is constantly active, leading to uncontrolled proliferation and the cancerous state. The cell is trapped in a permanent growth signal, #TAG6, membrane recognition by pathogens, and immunity.
Let's shift our attention to how the cell membrane is used not just for internal communication, but for communication with and sometimes invasion by the external world. Starting with the diverse ways neurotransmitters act, #TAG, neurotransmitters, signal diversity.
Neurotransmitters are small chemical signals that bridge the tiny synaptic gap between cells, and the type of receptor dictates the speed and the nature of the response.
So for skeletal muscle, you need speed.
For skeletal muscle, acetylcholine binds to the nicotinic receptor, which is a rapid, ligand-gated sodium channel causing immediate depolarization. Speed is necessary for quick motor response.
But the heart requires a different kind of control, slowing things down.
The heart muscle uses a different mechanism. The muscarinic acetylcholine receptor, which is linked to a G protein cycle. This G protein opens a potassium channel, allowing potassium to rush out.
Which makes the cell more negative inside.
Because the cell hyperpolarized, even more negative inside, which inhibits contraction.
And what about inhibitory neurotransmitters, like dopamine in the brain?
Dopamine acts indirectly via the CAMP cascade. It induces adenylate cyclase, leading to increased CAMP. This activates a protein kinase that phosphorylates a membrane protein, which ultimately increases the membrane's interior negativity.
So it makes it harder for the neuron to fire.
By making depolarization less likely, dopamine effectively acts as an inhibitory neurotransmitter. #TAG-TAGB antigens and lymphocytes.
The immune system relies heavily on specific membrane recognition using surface receptors.
When foreign antigens bind specifically to surface antibodies on lymphocytes, this triggers an immediate and profound physical change in the membrane architecture. The antigen-antibody complexes rapidly cluster together.
A process called patching and capping.
A visible process called patching and capping. Yes. This physical clustering then leads to internal membrane changes, including altered permeability to ions and molecules.
And this is the signal for the immune response to start.
This cascade ultimately triggers the lymphocyte to divide and differentiate, producing specific secreted antibodies to fight the antigen. #TAG-TAG-TAG lectins, sugar-binding agents.
Lectins are an interesting class of glycoproteins, mostly found in plants and invertebrates, that specialize in recognizing and binding specific sugar chains found on the cell surface.
Over 60 lectins have been identified, and they are critical tools in cell classification and purification. Structurally, they're usually multivalent.
Meaning they have multiple binding sites.
I have several specific sugar binding sites per molecule, This multivalent structure allows them to cross-link adjacent cells or induce receptor clustering on a single cell.
And this cross-linking behavior is often associated with disease states.
Researchers observe that lectin cross-linking between adjacent cells is dramatically more common in cancer cells than in normal cells, though the precise significance of this observation is still a subject of research. But they are invaluable for categorizing different cell surfaces based on their sugar code. #TAG D viruses and receptors.
Finally, let's discuss viral invasion, where the virus's specificity for a host cell is dictated entirely by surface receptor recognition.
Enveloped viruses, like HIV, influenza, or rabies, use protruding glycoproteins on their membrane coats to bind to specific host cell receptors.
And that binding is the whole game.
Binding is the key event, followed by internalization through membrane fusion or receptor-mediated endocytosis.
Let's focus on the mechanism of HIV infection, where the target is the T helper cell.
HIV targets T helper lymphocytes because they possess the CD4 receptor glycoprotein. The virus has two key glycoprotein subunits on its envelope, GP120 and GP41.
GP120 is the one that sticks out.
GP120 protrudes outward and acts as the initial binding site for the CD4 receptor. And structurally, the interaction between GP120 and CD4 is highly similar to how CD4 naturally interacts with its intended substrate.
So once GP120 has bound, what does GP41 do to facilitate entry?
After GP120 binds, GP41, which is an intrinsic transmembrane protein, penetrates the host plasma membrane. This penetration is essential for fusing the viral envelope with the host cell membrane,
letting the virus's contents in.
allowing the virus's genetic material to enter the cell and begin its lethal replication cycle. Current therapies, such as using soluble CD4 receptors, aim to block this initial GP120-CD4 interaction to prevent that fusion event.
To bring all these concepts of structure, function, transport, and signaling together, we turn to the ultimate model system for membrane study, the erythrocyte or red blood cell. Hashtag A, the model system.
The red blood cell is just ideal because its plasma membrane is its only membrane structure. Chemically, it's a robust structure, roughly 40% lipid and 60% protein.
And we see a clear asymmetry in the lipids.
A clear lipid asymmetry, yes. Choline phospholipids like phosphatidylcholine are predominantly external, while aminophospholipid like phosphatidylserine are internal.
We mentioned their biconcave shape earlier, which gives them flexibility, but they are also quite rigid compared to other cells.
That rigidity comes partly from the high cholesterol content, particularly in the peripheral regions of the disc. The cholesterol to phospholipid ratio is critical. Changes here can significantly alter cell shape, leading to adverse hematological consequences. Hashtag B membrane protein architecture. Figure 228, table 211.
When we look at the protein architecture, we see two distinct classes: the intrinsic proteins that span the bilayer and the extrinsic proteins that form the submembranous skeleton.
The major intrinsic proteins include glycophorin, which carries the MN blood group antigens but whose definitive function is still unclear, and the crucial transporter, BAN3.
And what does BAN3 do?
BAN3 is the essential anion transporter. It allows chloride and bicarbonate to rapidly exchange across the membrane, and it spans the membrane 6 times.
And the extrinsic proteins form the internal structural support system, the cell's scaffolding.
They form the submembranous skeleton. This network provides the integrity, elasticity, and the characteristic biconcave shape. It's a complex, electron micrograph visible structure composed of a hexagonal array of large, flexible, rod-like protein tetramers called spectrin.
And how is that massive spectrin network physically anchored to the membrane so it doesn't just float away?
The spectrin tetramers link non-covalently to short bundles of actin at junctional complexes. Additional proteins like band 4.1 and desin stabilize this spectrin-actin association.
But the real link is anchorin.
The crucial link to the bilayer is provided by the protein anchorin, which connects the spectrin scaffolding directly to the intrinsic band 3 protein. This structural skeleton is so vital that similar spectrin structures have been found in neural and muscle tissues, not just red blood cells. Hashtag CC. Clinical deficiencies, table 212.
Given the importance of that skeleton, structural defects inevitably lead to abnormal cell shape and disease, often resulting in the rapid breakdown of the red cells. The most common example is hereditary spherocytosis.
This is the most common hemolytic anemia in Northern Europeans. The fundamental issue is a significantly reduced level of spectrin, leading to a defective membrane skeleton.
So the cells lose their shape.
Without proper support, the cells lose their biconcave shape and become fragile, spherical cells, spherocytes. These fragile cells are quickly destroyed by the spleen, causing severe anemia.
And we see other defects affecting the shape itself, like hereditary elliptocytosis.
Here, the red cells are elliptical due to either a heat-sensitive or sequence-altered spectrin protein. The protein is present, but is structurally compromised.
And then there's ovulocytosis, which is fascinating because the defect is not in the skeleton proteins directly, but in their attachment point.
Right. Ovulocytosis involves the intrinsic band 3 protein, the anion transporter, acting too rigidly.
So it's not a problem with the skeleton, but how the skeleton is tied down.
Correct. A change in the cytoplasmic region of band 3 causes it to bind much too tightly to anchorin. This restriction in band 3's mobility effectively anchors the membrane components so rigidly that the cell is constrained into an oval shape. These examples show how minor flaws in the supporting structure, the binding protein, or the integral protein can all lead to severe red cell fragility and disease. Hashtag outro.
What an incredible deep dive. We started with the simple passive movement of water driven by thermodynamics, moved through specialized high-speed gated channels that fire our neurons, examined the complexity of ATP-driven pumps fighting massive concentration gradients, and finished with the sophisticated signaling pathways that underpin our senses and the structural scaffolding that holds our blood cells together.
It's a journey for sure.
The cell membrane is simultaneously a physical boundary, a chemical battery, and a complex processing unit.
The clinical takeaway for me is the profound structure-function relationship we've seen repeatedly. It's astonishing that a single tiny molecular defect, whether it's one amino acid deletion, preventing a channel gate from opening in cystic fibrosis.
Right, or a mutation in a receptor's cytoplasmic tail, causing the body to fail to internalize cholesterol in familial hypercholesterolemia. It's just amazing that these tiny changes can disrupt that highly tuned machinery and lead to devastating systemic disease. The membrane is often where health and disease really intersect.
It's astonishing that this complexity arose evolutionarily. I think it leaves us with something profound to ponder. Consider the deep evolutionary family ties between the sodium-potassium, calcium, and proton pumps, and how the single ancestral structure became essential for muscle contraction, nerve firing, and energy generation.
And just the dynamics of it all.
Exactly. And remember the dynamics. How does the fibroblast internalize the equivalent of its entire membrane hourly through endocytosis, and yet maintain its structural integrity by continuously recycling that membrane back out? That constant dynamic turnover and maintenance is the true marvel of the cell boundary. Thank you for joining us on this deep dive from the Last Minute Lecture team.